to highlight few basic things. Like PDGFRA, as all of you know, it stands for platelet-derived growth factor alpha. It is located on long arm of chromosome 4. It has got 4 domains, namely extracellular domain,
color is a bit
So it has got 4 domains: extracellular domain, transmembrane domain, juxtamembrane domain, and 2 tyrosine kinase domains. So what is most important here is that the juxtamembrane domain, which is located in exon 12, it has got autoinhibitory effect on tyrosine kinase. So and one more important thing is that this region is more prone for breakage. That's why the breakpoints are more common in exon 12, that is in juxtamembrane domain of PDGFRA. So this schematic representation depicts the mechanism of formation of FIP1L1 PDGFRA fusion gene. PDGFRA and FIP1L1 both are located on the long arm of chromosome 4. In between you can see here the CHIP2 region. So because of an interstitial deletion spanning 800 kb region which involves CHIP2, these distantly placed FIP1L1 and PDGFRA, they come in closer proximity, leading to the formation of fusion gene. So not only CHIP2, but few sequences of FIP1L1 and PDGFRA are also lost due to interstitial deletion. So ultimately, the initial 233 amino acids of FIP1L1 fuses with the last 523 amino acids of PDGFRA. What is important here is that with this interstitial deletion, as I said earlier, we will be losing the part of juxtamembrane domain, which has autoinhibitory function against tyrosine kinase domain. So as it is lost here, it leads to constitutive activation of kinase domain. So the persistent eosinophilia in these cases. But thanks to the magical drug imatinib, which can cause treatment-free remission in these cases. Not only FIP1L1, apart from FIP1L1, we have got 18 more partner genes associated with this fusion. So ultimately, whatever the partner gene may be, it would.
Can you open the
previous slide.
Okay, now this is one of the techniques where we use breakup of fish to identify a fusion. Actually, breakup of fish you use to identify breakup of right? But here we use here is a condition where we use a breakup of fish to identify a fusion. Because traditionally we use breakup of fish to look for breakup of fish.
Yeah, I'm going to show that slide also. Maybe there you can explain. So apart from FIP1L1, we have other partner genes which will also bind to the same exon 12 juxtamembrane domain, leading to loss of autoinhibitory function of juxtamembrane domain. The same thing was also confirmed by experimental CRISPR-Cas9 gene editing technology, which confirms the deletion mechanism and the oncogenic potential of the fusion. Same thing can be viewed on integrated genomic viewer. In the center, you can see the black lines. These indicate the deleted region. So maybe you can talk about this.
Good. I thought you directly go to NGS.
Sorry, sir?
I thought you directly go to NGS. Fine.
We do fish also. So as I have already told you, all these 3 genes are located in close proximity at 4q12. You can see here. When you zoom into it, you can see that FIP1L1 is there at one end, CHIP2 is there in the middle, and PDGFRA is there after that. So here we use tricolor probe to target three regions of interest. We use orange probe to hybridize CHIK2 region and spectrum green to hybridize PIP1L1 and proximal region. Spectrum aqua to hybridize PDGFRA and the region distal to it. So normally you know that each gene has two copies. So normal cells should have 2 blue, 2 green and 2 orange signals. So as they are in close proximity, you can see it's a fused signal. But whenever there is loss of CHIK2 leading to fusion formation, there will be loss of CHIK2, so loss of orange signal is expected. That is what's shown here. There is only CHIK2 loss, so there is no orange signal. In the normal cell, you see 3 colors fused together. So, PIP1L1 PDGFRA fusion, why it causes preferential proliferation of eosinophils is largely unknown till date. But few papers are coming up talking about JAK2 as a mediator of PIP1L1 PDGFRA induced eosinophilia. So basically, PIP1L1 PDGFRA it activates STAT5 and STAT3. But STAT5 activation happens by some unidentified kinase. The kinase is not known. Only the STAT3 enough kappa beta activation happens through JAK2 dependent pathway. So JAK2 it activates PIK3 signaling cascade also, and via STAT3 enough kappa beta pathway it expresses c-MYC and survivin genes. They are target genes for JAK2, by which it causes eosinophil proliferation. So if you try to use JAK2 inhibitor, you can only suppress this pathway, STAT3 pathway, but the STAT5 pathway will remain untouched. Next, coming to the clonal evolution. Now, so far everything is fine, like how PIP1L1 PDGFRA causes downstream pathways and further proliferation of eosinophils and all. But in due course of the disease, due to selective pressure, due to TKA therapy, or due to emergence of PDGFRA resistant mutations, or due to clonal cytogenetic abnormalities like trisomy 8 and 12, it can transform to blast phase. All these are all well documented and known. But what is unknown is the epigenetic regulators, the role of epigenetic regulators such as BCOR in our case. So I'm just reminding you that along with PIP1L1 PDGFRA, we have also reported 3 BCOR mutations with low VAFs. Just remember this point, I'll come to this later. So before going into BCOR, we'll just try to understand the normal function of BCOR. Do you know what BCOR stands for? BCOR stands for BCL6 co-repressor. Why co-repressor? Because it cannot directly bind to DNA. As you can see in the picture, it is not directly binding to DNA. It can only bind to some DNA binding proteins such as KDM2B. Okay. So the normal, what is BCOR? Is it tumor suppressor gene or oncogene? Sorry? Tumor suppressor. So what is the function of tumor suppressor gene? It has to suppress gene transcription, right? So it has to suppress front benchers. So it has to suppress gene transcription. So the process of suppressing gene transcription is called repression. So for which we have a few complexes in our body called repressor complexes. In mammalians, it is named as PRC. They resemble home, so that is called polycomb repressor complex. Okay. So what I mean to say is BCOR is always associated with this PRC complex. So via KDM2B, finally it gets attached to your DNA. So in this position now, with the complex and cofactors in hand, BCOR can cause monoubiquitination of histone proteins like H2A, which in turn cause recruitment of SRM, which is a component of PRC2, that is polycomb repressor complex 2. So which ultimately leads to trimethylation of H3 histones. Okay, so all these histone modifications are happening. So this will ultimately cause gene suppression. So BCOR is a negative regulator of Hox genes and SEPA family. You might be knowing that Hox gene is important for myeloid differentiation. Now BCOR is trying to regulate myeloid differentiation and proliferation. This is the normal function of BCOR. So what are compound mutations? Can anybody guess? Try to define what are compound mutations. Anything, whatever comes in your mind, you can happen.
Still not hazard.
So compound mutations can be defined as multiple mutations happening in a single gene in a single neoplasm. Now can you give me an example? Seeing our case, I just showed you one mutation here. So multiple mutations happening in a single gene in a single neoplasm. So this is a classical example of compound mutation. So whenever you see BCOR mutation, you tend to see multiple BCOR mutation. The reason being, if you try to recollect multi-hit hypothesis or two-hit hypothesis of tumor suppressor genes, why does it happen, two-hit hypothesis? Because to nullify the effect of tumor suppressor genes, you need to inactivate both the alleles. Then only the tumor suppressor activity will be suppressed. Okay, so here also, compound mutations in BCOR will profoundly cause loss of BCOR activity, driving the clonal expansion, impaired differentiation, that means increase in the blast, and enhanced lipomogenic potential. All these are contributed by BCOR mutations because it has lost its activity because of compound mutation. In simpler terms, by biallelic inactivation. So all these are good. So I have explained to you like FIP1L1 PDGFRA mechanism, how it causes eosinophilia, and what is the normal function of BCOR, and you are like almost convinced that BCOR mutations will cause blast crisis in this case. But is there any evidence? So could you find any evidence?
You didn't look.
No. So it's a big no. There is no direct evidence stating that BCOR co-mutation in PDGFRA driven myeloid neoplasms will cause blast crisis. That is not there. There is no direct evidence. But there are multiple papers which states that BCOR can cause leukemic transformation in CML cases and also in pH negative myeloproliferating neoplasms. So let's see one by one. This is the paper published in Blood in 2013 by Dam et al. So they used to wonder why low risk MDM patients are rapidly progressing to AML. So then they started doing whole exome sequencing on those patients who presented with low risk MDS but rapidly progressed to AML. So they have found that those patients with low risk MDS, they had BCOR mutations at a low VAF, as we see in our case. So what they have concluded is that the study suggested that BCOR mutations contribute to disease progression rather than initiating the disease. Because if a mutation is initiating the disease, it would present with high VAF. If it occurs at the later part of the disease, it would present with low VAF. This is the difference. So they have also suggested to include BCOR analysis in considering risk stratification. This was in 2013. Now we see in 2022 ELN classification, BCOR is classified under adverse risk category. If you see BCOR mutation, it be like MDS or AML MDS or AML, it is adverse risk. There is one more paper published in Blood in 2021, BCOR gene alterations in hematological diseases. The authors say that BCOR mutations are very rare in chronic phase of CML, but when present, they usually persist despite being treated with TKI therapy. So which tells us that it could be originated from a pre-leukemic pH negative clone which had existed independently along with the pH positive clone. It was a CML case I am talking about. And they have also said that BCOR mutations have contributed to driving CML transformation in those cases, and they also say that BCOR and ASXL mutations, they appear to be independent predictors for worst response to TKI in BP phase of CML. So again the same thing, clonal evolution and clinical implications of genetic abnormalities in plastic transformation of CML. Even here the authors suggest, like they have found that frequently found mutations were all these genes, one among them was BCOR. Again one more paper in 2023, ELN lab recommendations for the diagnosis and management of CML. They have also found BCOR in almost 15 to 20% of cases of CML BP, that is blast based. So not only CML cases but Ph negative embryons were also found to have BCOR mutations when they transformed into blast crisis. So based on these literature review and findings, now we can talk about impact of BCOR on disease biology and prognosis. So as you have seen, BCOR mutated myeloid neoplasms, they are enriched in high risk disease categories and they are associated with poor prognosis. Loss of BCOR disrupts normal hematopoietic transcriptional programs leading to leukemic transformation, and compound mutations further aggravate the disease phenotype. BCOR mutations, we have seen that they were commonly studied in AML, MDS and blast crisis of Ph positive as well as Ph negative MPN. But there is no direct evidence or no paper till now talking about the impact of BCOR in eosinophilic neoplasms with FIP1L1 PDGFRA fusion positive cases. So since FIP1L1 PDGFRA fusion defines a distinct molecular subset now under WHO 2022, and it also responds well to targeted therapy, but the presence of BCOR compound mutations could theoretically modulate disease biology by contributing clonal evolution or progression, which we have already evidenced now. So to conclude, this case exemplifies the complexity of diagnosing and managing myeloid neoplasms with PDGFRA rearrangement, especially in blast phase and in the presence of mutations conferring drug resistance like BCOR compound mutations. So targeted therapy with imatinib remains the cornerstone, but emerging molecular insight necessitate personalized approaches and clinical trial participation. So in this case, as sir has already mentioned, initial marrow was sent to cytogenetics fish as well as NGS. We have found these compound BCOR mutations in initial marrow, not in the marrow with the blast crisis. So that's why I have written this point to highlight the significance of molecular testing. So biologically, genetic mutations occurs first, obviously, and they initiate the disease process. Of course, here the disease progression it has initiated, while the characteristic morphological changes become apparent later as a consequence of these underlying molecular alterations. So we could get the patient sample at the verge of transition. So patient had already developed compound mutations in BCOR, and at the verge of transition we have got the sample. Luckily we could find the BCOR mutations which are like important for leukemic transformation and blast crisis. After a week the patient worsened and they could find increase in blast and morphology. So the genetic lesion could happen first and then only it would be manifested in morphology. So this is the classical example for it.
Second sample you didn't get BCOR mutation?
No.
We didn't do it.
That was a second repeat sample you are asking? No, they have not said.
I think as you
Repeat
can,
marrow was not said.
2 NGS within 2 weeks with the leukapheresis, ICU stay.
That was one question.
Yeah.
Like, if we
But
have
despite
had this
being BCOR positive, still you continued with tyrosine inhibitor.
So, you mean the first, if I had known that